Hi, and welcome back to the wonderful world of the scientific hypothesis. My name is Brad Alger. You may have heard that science is suffering from a reproducibility crisis. That is, researchers can't always duplicate or reproduce each other's experimental results. However, reproducibility is a core value of science. Ideally, all research findings would be reliable additions to our scientific knowledge and could therefore be confirmed and repeated by everyone. But science is extraordinarily complicated, and this ideal of reproducibility is impossible to achieve. So, does this mean that we face a crisis? In the next few videos, I'll look into a few details of the problem. The first thing to do is to define exactly what it is that we mean by reproducibility. In this video, I'll show that several aspects of an investigation may be subject to a reproducibility test, and you'll see that not all failures are cause for alarm. We'll need to sort these things out in order to put the talk of crisis into perspective.
To introduce the topic of scientific reproducibility and whether it's a crisis or not, we'll first have to discuss what reproducibility itself is. We'll look into the question of what is it that's reproduced, and as I've mentioned, there are several different aspects of an investigation that may be subject to reproducibility tests, including analysis, the data, and the conclusions. And finally, we'll get to the question of why this all matters and how it affects our understanding of science. Now, there are many reasons that reproducibility is important to science. Here are some of the key ones. First, science relies on objective observations. In order to be objective, the observations must be repeatable by others. Science seeks true explanations, which make predictions that are reproducible. Almost by definition, a true explanation has to be one that can be seen by other people. And finally, reproducibility helps to define science. Science can't study miracles that can't be reproduced. We can distinguish among four classes of reproducibility studies according to what it is that's actually being reproduced. In the first case, the original data is being subjected to a reanalysis, and this gives rise to analytic reproducibility. In the next case, the exact experimental conditions are duplicated to see if the original data and conclusions follow, and this is called direct reproducibility. If the general conditions are maintained, but some specific details are allowed to vary, then this can give rise to systematic reproducibility studies. Finally, if the general scope of the original hypothesis is being tested with an entirely new set of predictions and experimental conditions, then this gives rise to studies that are called conceptual reproducibility studies. I'll give examples of each one of these and discuss them in a few minutes. But I also want to point out that throughout, a theme will be to examine whether or not these different cases of reproducibility studies should actually fall under the category of reproducibility. And as you'll see, in two cases, this seems to be a valid characterization. However, in two other cases, this seems inappropriate, as I'll explain. To provide some context for the discussion, let's consider the case of a specific hypothesis. We've perhaps observed differences in the cognitive behavior of animals, mammals in this case, and we've noticed they eat different foods, and we've formed the hypothesis that diet affects the cognitive behavior of mammals. This makes a specific prediction that rats, for example, fed a special diet will learn to run mazes faster than rats fed a standard blah diet. To test this hypothesis, we'll compare maze learning ability of two groups of rats as alike as possible. We'll give one group rat high Q food and the other the blah food. We'll place the rats in identical mazes and we'll record the time it takes them to learn to run the mazes. Now in this case, our data indicate that the rat HQ food seems to improve the performance. Statistical analysis shows a significant difference, and this leads to the conclusion that data are consistent with the hypothesis. So now we assume we've done the experiments as just described and published our results. Another group comes along and wants to do a reproducibility test, and this group elects to do a test of analytic reproducibility, and that means this group will repeat the analysis on our data to determine whether or not the analysis of that data actually supports the original conclusions. So this is analytic reproducibility. Another group attempts to repeat our procedures exactly. Replication of the original experiments as much as possible, trying to see whether or not the original data can be obtained. So the subjects are the same, the treatments are the same, the tasks are the same. Hopefully the data will be the same, that's what they'll be looking at, and determining whether or not the statistics and the conclusions follow along. So this is a case of direct reproducibility. Now let's assume that another group comes along with a slightly different question. This group wants to know if varying the original conditions somewhat support the original conclusions. This would be a case of systematic reproducibility. Now, in this case, they may switch and use mice. Other treatments would be exactly the same. It would be the same food, the same task, and so on. The question is, if one condition is changed, would the conclusions follow as they had in the original case? Finally, we can consider the case in which another group decides to test the overarching hypothesis more broadly. You recall the hypothesis was that diet affects the learning behavior of mammals. This group then elects to test an entirely new set of predictions to determine whether or not the new data would be consistent with it. These new predictions might involve changing all sorts of conditions, including the subjects, the treatment itself. Notice different foods are now involved, as well as the task, no longer the simple maze task, but perhaps an entirely different sort of behavioral learning manipulation. What's at issue here in examining all these changes is to determine whether or not the fundamental concept underlying our original conclusions was valid. So, a quick review of what we've said. If the focus is on data analysis or the experimental results themselves, then these cases are genuine instances of reproducibility studies, and they're called analytic or direct reproducibility. In these cases, genuine difference in results obtained by different laboratories, this is a potentially very important issue for science. Genuine problems might exist. However, in cases that have been called systematic and conceptual reproducibility, nothing is actually reproduced. In the first case, certain conditions are allowed to vary before the tests are made. In the second conditions, entirely new predictions of a hypothesis are the focus of attention. The attempts are being made to extend the original findings, not to replicate them, and therefore these should not be considered as reproducibility studies at all. Regardless of the outcome, these studies are not a problem for science. In fact, if you think about it, it's clear to see that the heart of experimental science is testing for systematic or conceptual reproducibility. These tests challenge and potentially falsify hypotheses. Their outcomes move science forward. Misunderstanding this obscures the nature of the reproducibility itself and misunderstands the fundamental goals and operations of science. I'll come back to some of these issues in later videos. From now on, I refer to direct reproducibility and not the other classes. Thanks for watching. Remember to give it a thumbs up if you like it and subscribe to hear more. See you next time.